Okay, welcome to the afternoon session. So we'll start with Glen Dsouza from ETH.
Right. Hi, everyone. I hope you had lunch, and in the next 20 minutes, I had a perfect line, but I'm going to go with it anyways. Next 20 minutes, I'm going to convince you how what bacteria eat influences who they interact with, and how do they get how does this impact nature. Right. So this is work largely done in collaboration with two postdocs, Johannes Kijkstra and Julia Schwarzman, ETH Zürich and MIT, and Martin Ackermann, who's also my postdoc mentor. So if you think of what bacteria grow in natural environments, it's not often simple sugars, right? Not glucose, xylose, etc. It's often very complex bits of polymers. If you look at, for instance, satellite image of a Baltic Sea, this is just off the coast of Sweden, you'll see a lot of these green, and this is essentially alga. And these algal cells often throw out very complex polysaccharides into outside their cells, and this is essentially what bacteria feed on, especially in the ocean. And this is easily applicable to, say, bacteria that grow on, in the soils. This is possibly like dead trees, or again polysaccharides like cellulose or xylan. And these polysaccharides are abundant depending on which paper you like. It's between 40 and 95% of all the carbon or the bioavailable carbon out there in nature. And if you're looking at, say, largely marine environments, this could be chitin or alginate. If you're looking at terrestrial environments, as I said, it could be xylan or cellulose. And what bacteria often do, not all, but most, is they exist. They throw out a lot of extracellular enzymes with which they can chop up these complex polymers into oligo or monomeric bits. Now, why is this relevant? This is because not all bacteria can do this, some can do that. And essentially, if you look at it in the context of an ocean, this activity essentially propels the carbon cycle, right? So you have a lot of CO2 being absorbed, fixed by largely phototrophic organisms. This ends up as marine snow, if people would call that. And this is by heterotrophic bacteria's degradation events, this enter the carbon cycle and make it available to many more organisms or trophic levels. And if you simply go and ask, do an experiment in nature where you now have alginate, a very common polymer that alga produce, and incubate this in water. You just take a patch of a liter of marine water and incubate that with alginate. What you'll see is a lot of functional ecotypes or genotypes that you would often find. So you'll find these alginate degraders. Now they have the enzymatic capacity to degrade this alginate. This produces oligoalginate, as I said, and this is available to the alginate degraders to grow. And as a consequence of their growth, they also produce a lot of byproducts. Now, these are not the only players in the system. You'll often then find types that can perfectly exploit what's out there, right? So you'll have the cross feeders, which can eat oligomers. We call them often scavengers. So they can just do this. They don't have any of the enzymatic capabilities, and you can have cross feeders that can't do any of these steps, but will largely feed off whatever byproducts is out there. And these sort of simple ecosystems are pretty replicable across different environments. And this is something we leverage to study microbial ecosystems in the lab. And one system we use is simply 3 different bugs that came from marine environments off the coast of Massachusetts. These were isolated by Otto Cordero and Martin Paul's groups. Essentially, there are 3 players that I will talk throughout this talk. So that's Cyclotrophicus, which can degrade alginate, Vibrio tasmanensis, which can eat oligomers, and then Rogeria species, which can eat these byproducts. And one question that is, how do these ecological interactions manifest in these systems? And as an ecologist or microbial ecologist, what you would often do is just do pairwise or co-cultures or monocultures, right? So this is what the first step from us for developing the system. So what I basically do is I just look at growth yield. This is a function of how much inoculum was there at the start and the end of the experiment. And this is an alginate degrader when alone, and this is an oligomer consumer when alone. Now, if I do this experiment in, say, co-cultures, where I'm now pairing both of them in the same flask and then measuring how many what's the growth yield of either type, and this is simply to these cell types have different morphology, so it's easy to do simple plating and discern these types. So we find that we do know that cross-feeding interactions should benefit the oligomer, produce oligomer consumer, but what we also find strikingly that the degrader is also benefited by the presence of these oligomer consumers. Now we have another play in the system. I can do the same experiment. Degrader grows as usual. The byproduct cross-feeder doesn't when alone. Now if I do this in say pairwise co-cultures, the degrader seems to be not benefited or not affected in this case, but the consumer is clearly benefiting, right? So what this is showing you, the essence of this is that cross-feeding interactions don't just propagate downstream, especially in this case, they can have upstream effects where consumers can have some benefit on the cross-feeder, on the primary degraders in this system. But this is, again, pretty well mixed situations, you could argue, not reminiscent of the wild. And that is because, I don't need to convince this crowd, but needless to say, we do know that most of microbial life is often spatially structured, right? So it's imperative that we understand these interactions, taking into context this spatial structure. And one consequence of this spatial structuring is there's a lot of microscale interaction. So cells exist in very close proximities in each other, and it's essential to ask those interactions. And then it's also becomes important to ask which types of interactions, right? So there can be metabolites shared, and these can have, depending on what chemical compounds are exchanged, can have antagonistic effects on the coexisting strains, or you can benefit, as is seen in this question. And the question really we're interested in then is like, so what is the consequence of these interactions on, say, community functions? And for us, community functions is, in this context, is very easy. We just ask in terms of remineralization of carbon. And one metric that we sort of use is, for this remineralization of carbon, is the influence on growth and behavior of cross-feeders on the degrading type. And these are the only ones that can do all the heavy lifting in the system. But first, we need to be able to study these interactions at the microscale, right? And for that, we leverage something that is predominantly used in a group, which is microfluidics. And long story short, it's essentially very small devices where you could think of, that we essentially build thousands of houses where cells can grow and survive, right? And we provide them with nutrient A. We can keep on triggering different nutrient conditions in this thing, so we can grow them at X and then change it to B. And lateral to these feeding channels are these small houses, or we call them family machines, where cells can grow, divide. And then using automated microscopy, we can essentially monitor the behavior of cells as they grow in space and time for very long, say, at least for long ecological scales. And then what you often get as a consequence of these images is, if you're using epifluorescence, you'll probably see this, cells growing in time and space. But we want to be able to go from just looking at growth to having a quantitative, say, measure of this growth. So for that, we develop quantitative analysis pipelines, which is essentially, it's a neural network which has been thought to identify cells and find objects in these time lapses. And then using lineage tracking, we can basically assign identities, or we can get measures of growth, right? So in this case, every cell type that has the same color arises from the same lineage, right? So they're essentially part of the same family, or in other words, they are originating from the same founder cell in this system. And so with this, we can essentially do experiments over long ecological scales and then have a quantitative estimates of who's there, who interacts with whom, how fast they grow, etc., etc. And so with this, then we can start asking a question we had, which is, how does the presence of cross-feeders impact behavior of the degraders? So one measure we have, so if we grow degraders alone, this is often what you'll see. Post-division, degraders tend to stay next to each other, so they engage in this, what we would call aggregative behavior. They tend to form these small microcolonies, right? And now we can ask how this behavior changes as a function of who's there. So now we can do the same experiment with the oligomer cross-feeder. So it only eats the oligomers, it doesn't eat the, cannot degrade the alginate, so it has to rely on supply from them. And as you see, there's a slight bit of difference. The oligomer consumers in cyan don't grow a lot. Now, into the same experiment with the byproduct consumer, you can see here there's a little bit of, there's a much bigger difference, where there's very rarely very little growth events or division of the alginate degraders. So what this is essentially saying is that yes, if you are coexisting with a cross feeder, you can effectively change some sort of spatial behavior of the degrader strain or the primary producer in the system. And depending on which type of cross feeder is in the system, there could be differences in the benefits. Now we can go out and quantify what the magnitude of these differences are, right? So one simple measure is the number of resident cells of the degrader as a function of which the community context, right? So if it's alone, it's the orange line. If it's with the oligomer scavenger, it's pretty similar in terms of how many resident cells stay in these chambers over time. And we can look at multiples of chambers. However, if we now look at, which is evident in the image, if we pair it with the exploiter, we see very few cells. So there's a little bit of effect. Now we can also ask, we can forget the numbers, we can ask as a function of, say, spatial distribution. So we can look at every cell in this system and ask what is the average amount of cells that exist, say, in a given unit of distance. So this is what we call as intercellular density. And we have a measure, which is approx, like if it's alone, there's about 20 cells per micron in this image. However, if you introduce a scavenger that, from a well-mixed situation, we know that benefits the presence of the degraders, you actually increase the spatial, how many cells are in one given unit. And if you add another type which scavenges this, which utilizes the byproducts, you then reduce this. And this was already in. So I think what essential conclusion here is, if you have interactions, you do change the spatial distribution of cells in a system. And this can allow us to now ask how do interactions influence, say, spatial organization in, say, more natural situations. But I think what we also, as I said, because we're interested in community functions, we're interested in understanding the growth of the primary type as a function of who it's cross-feeding with, right? So what we can do, because we've looked at many cells across long time scales, we can look at growth rates. And how do we measure this? Is pretty simple. Because we're monitoring cells at different time points, we know what their cell lengths are, or we know what their cell area is. So if you just plot length, it increases and declines. And this is pretty similar to, say, looking at growth curves, right? So we can do change in length over time for multiple such cells, and we can then build growth rate distributions, right? So if this is such a distribution for, say, when an alginate degrader is in monoculture. So this is different time points in the experiment, and we can see that the distribution, so this is the median of the distribution, and it's sort of like if you have degraders alone, they are pretty much growing at maximal growth rates from the time they arrive in the system. Now, if you pair one of the oligo consumers, you essentially shift this dynamics, right? So the cells take longer to reach their maximum growth rates, or even they often fail to reach growth rates that they would achieve when they are present alone. And this becomes even more dramatic if you have, say, another kind of consumer in the system. So what essentially tells you is that cross-feeding not just changes spatial distributions, but it can also influence the growth rate of these primary degraders in this system. And so it's important to sort of incorporate this. And these effects are not always downstream, which is often the case with our measurements. It can also often be upstream effects in these trophic chains. And we can also do the same experiment. We can look at growth rates of the consuming types in this system. And one thing that is pretty evident is this is super low, right? Like they're effectively dividing once every 10 hours or so. So what it's telling you that cross-feeding, although prevalent, might not be the optimal way for cells to depend, especially in systems where you have to rely on someone for your metabolic supply. And this is going to be a problem, right? So how do cells solve this dilemma? If they have to cross-feed, how do they increase the propensity that they get these nutrients? And when we look at most of these genomes, or some of the genomes of these consuming types, they seem to have, which some of you might know this, is a type 6 secretion system, right? And essentially what this is it's like think of it like a spear gun with a poison tip, and cells can just punch holes into their neighbors. And a result of this punching holes is the release of metabolites, right? And if cells have this can be a pretty neat way for cells to acquire nutrients in, say, situations where you're in close proximities of each other. And this is what we then sought to test. And I'm giving you small teaser for a talk that will come up in 2 hours from Astrid. So I'm just going to let you stay with the image and think about questions for Astrid. Right. So what we can do is we can take a degrader and we can take another crossfeeder, which we know has a type 6 system in it, and we can pair that. And when we do, sorry, this is an analogous system. I forgot to switch the movie, but same principles. Think of this as a degrader. Think of this as a crossfeeder, except we've now labeled the type 6 secretion system, right? So you can see initially the system is flush with degraders. That's the magenta type. That's because the green type cannot grow as fast. And now if we let these systems play, you can see a lot of these lysis events, right? There's a lysis event here, and cells essentially are punching holes, taking up nutrients, and growing in coexistence with these degraders. And this is how cells, this is one way for cells to solve the cross-feeding problem they face in communities. So with that, yeah, so stay tuned for more, right, at 4:50 for Astrid. So with that, I would like to thank my cross-feeding system that often has problems staying or leaving. And 5 of us are here, so it's essentially 30% of the lab is in Cambridge right now. So feel free to reach out and ask more questions. And also thank my collaborators who help, and then funding sources. And you guys for attention. I'm happy to answer any questions.
Thank you for great talk. Questions? I guess I can, oh, yes, please.
Have you tried simulating the type 6 system?
Sorry. Can you repeat the question?
Have you tried simulating the type 6 interactions?
Simulating in terms of modeling?
Individual bit, like an individual based model.
I think Astrid, that's the next talk. That's in 2 hours, I guess. Yeah.
Orkan?
I was going to ask about the sort of the environmental aspect of this. The degrading enzymes, is there any idea whether they get dispersed or in the sort of the ocean environment?
So if we randomly would sample a bunch of cell genotypes, what you would find is there's the cell types which secrete a lot of enzymes, and there's cell types which would often keep them on the cell surface. So these enzymes basically would be tethered. So you could have sort of broadcasters of enzymatic degradation, and you could have private degraders. So you can find both. There's also evidence for new types of cell types which would actually chop off the polymer inside the cell one by one. So there's very little loss to the external environment.
Yes.
Yeah, thanks for your talk. Maybe I got this wrong, but in the first experiment that you showed, I think you showed that there was a positive impact on the degraders.
Yeah.
But this was not the case in the validation.
In terms of growth, no, exactly. So there was the well-mixed situation was not reminiscent of actually like this microscale situation. Yeah.
And the first experiment was this done on plates?
This is just shaking flasks. Exactly. Yeah, exactly. I think it was pretty much like a source sink. These cell types are degrading the polymer. There's simpler types accumulating, and it's obvious the simpler types could slow down a reaction. And if you have a consumer which takes that, you keep that reaction on, so you break down much more. So I think that's the easy explanation. No, we haven't, but we've been thinking of doing it on, say, agar plates with alginate in them. So yeah, but we haven't done them.
Any more? I guess I'm quick. Oh, yes, please.
Nice talk. I had a question about, you used the shape of the cells to track actually how they're growing.
Yeah.
Are they actually changing their shape? Growing faster?
So these vibrios don't. Some of the vibrios do. They become smaller when they grow faster. These ones largely keep the same. The degraders at least keep the same cell shape. Oh, stay with it.
Yes, more.
In some of your initial microfluidic videos, there seems to be quite a lot of loss of bacteria sort of during the time, rather than sort of keeping all of them within the system.
Yeah.
How do you account for that when you're keeping track of growth rate?
So in this, it's a very stringent cutoff. They need to the same cell or in the same should be in about the same spatial location for about 5 frames. So about.
What happens to the ones that haven't been in there? They grow and then
So if they're leaving, we basically ignore them. we're basically looking at, in this context, we're just looking at the number of cells that were resident for a long time in the chamber. But yeah, there's about 20% of cells that decide to leave. So if so, one of the triggers we know they leave is because if they sense there's too much of breakdown products around. So for them, so this is with this another strain, we basically can stimulate leaving by introducing simpler breakdown products or increasing cell density. So I think this sort of uses as a cue. If there's a lot of simpler products, it's probably a competition sign. So the growth rate actually declines if they're simpler ones. So I think they're sort of balancing these things.
Okay, the next, thank you. Let's thank Glen again. So next speaker.